name is Dahlia Bolt. I'm from New York University in New York City, and I'll be talking about using PAOs, polyphosphate accumulating organisms, and other microorganisms as essentially a nutrient timed release in fields for maize. So one of the major problems we're facing in agriculture is P shortage, phosphate shortage. So especially maize, they uptake phosphate during the kernelling phase, which essentially is producing corn. And if they don't have access to adequate phosphate, then you lose those kernels. You're not going to have great yield. But what you would think is that adding excessive fertilizer on the field during the kernelling phase would help. False. Only 20 to 30% of the fertilizer we apply is actually utilized. So everything else, all that, like 70% of your money is gone. It runs off and it creates pollution, freshwater streams are ruined, etc. It's bad. And to make matters worse, almost all phosphorus is sourced from minerals, meaning that we mine it. And within the next 50 to 100 years, we're expected to completely run out. No more. So what do? Well, we're looking at using microorganisms that cycle phosphate in soil, rhizosphere. The rhizosphere is the region of soil with microbacteria, with microorganisms that deal with nutrient cycling. And we're looking at essentially using polyphosphate accumulating organisms, PAOs, to cycle out ortho-P, the usable form that is used by plants. Now, poly-P is the polymer produced by this. So we've seen in wastewater, believe it or not, that these bacteria are able to remove phosphate in wastewater streams. So why not for soils? That is where we're searching now. And we've so far have done 7 different maize genotypes, and they were grown under high and low nitrogen conditions. And the reason we did this under different conditions is we're trying to create a very broad field conditions. We're trying to explore under what conditions do PAOs best thrive, what species communities are there, etc. Because just because we see like the same abundance of PAOs doesn't mean it's going to be the same type of PAOs. That's something we're interested in exploring as well. So we take these corn. So far, we have time point 1 data sampled, and I'll get to that in a moment. We take the corn and we sample the rhizosphere for the bacteria. Different labs have different definitions of the rhizosphere. So what we consider the rhizosphere is the soil directly attached to the roots. So to take samples, we take the corn roots, put it in a bag, beat off the soil. So the soil is only those directly on the roots, not just the general area. And then afterwards, we process the samples. So believe it or not, we're washing dirt, as counterintuitive as that sounds. And we're removing the bacteria from the dirt. And the reason we're doing this is inorganics can damage machines and downstream sequencing. How many of you guys have worked with fluorescence activated cell sorting?
Not yet.
Okay, it's a very finicky machine. It's a pain. So after we wash off all this dirt, we fix the solution. And the reason we fix it is we essentially kill the bacteria without destroying it. And we have to do that because this is a stress response, or it's a normal behavior. We don't want stress responses. So we're looking for like the phenotypic evidence, not genetic evidence, because just because you have the genes to do something doesn't mean you can do it. So putting a sample in a test tube stresses up bacteria so much it starts to act like a PAO. We don't want that. It's a false positive. Or if we have anaerobic conditions where there's no air in the soil, it starts dumping all of its phosphate, and now we don't have a PAO. So we fix it to kill it. Then we add a solution to make it so we can stain it. We use a fluorescent stain and put it through FACS, fluorescence activated cell sorting, and sort out the communities. And then once we have all of our sampling, we plan on doing 16S in sequencing to identify known communities and unknown communities. Now here's where things get interesting. As you can see from this FACS data, we do the gating to eliminate interference with autofluorescence and fluorescent spillover. So that's why we only have like the top, like the most fluorescent ones that are really showing up that they can do it. And as you can see, we see that there's a huge shift in community. There are PAOs in the rhizosphere, which is good. And we can see that there is a distinct shift. Interestingly, for PHA, we can see that there's no just difference in poly-P accumulation. However, there's a large discrepancy in PHA accumulation, with over 70% of PAOs accumulating a high degree of PHA. So if you take up if you make poly-P, you're going to make PHA, which is interesting because that goes against like what you think for this diagram, where it should be one or the other. Now, we also can see from our corn, this is time point 1. We don't have a trend yet because we only have time point 1. Corn takes a while to grow. We got to do this. And like to get the samples, you have to dig up the corn. It takes all day to do it. We can see that the bulk soil, which is the soil that doesn't have any plants growing in it, actually has the highest relative abundance of PAOs, which is not what you would expect for something that's crop cycling. But you have these two the H99 high N, and then the H99 NRT knockout, which essentially makes it so they don't uptake nitrate. It's nitrate stress. Low N and high N, highest on PAOs. So it seems like there's no like distinct difference between like how high and low N is across genotypes, but there's a huge difference in between the same genotype. We want to keep looking at future time points and see if this continues down the trend. Any questions?
Such interesting research. It sounds like you've been really busy this summer. And yes, I work with maize as well, and I know how annoying it is and how much you want to be able to look at more corn, but you can't because it grows so slowly.
Yeah.
One question, how do you see this impacting our current agricultural landscape? Because this is some fantastic technology, and I can see some real world applications for this. Would you be able to share some with our audience?
So I'm going back to the sewage stuff a little bit. I know this is plant research, but a lot of this stuff came from like sewage research. There's been talk of using PAOs to filter out phosphate from eutrophication water streams. So what if we did that in soils where like you add, like not necessarily add, but you create favorable conditions for these bacteria to thrive, and you use the bacteria as an auto release. So aerobic conditions in soil is like dry, sunny day. Anaerobic is like when it rains, there's very little oxygen, it's saturated. So you can have a timed release of phosphate based on weather conditions. You apply your fertilizer when it's dry, and then it releases when it's wet. It can be an automatic release. That's the hope for this, at least.
That's amazing.